Welcome to Pharmaceutical Technology's podcast, Trends in Bioprocessing Analytics, Weighing and Scale Up. This podcast is brought to you by METTLER TOLEDO, a global leader in industrial analytics, laboratory equipment, and industrial weighing for pharmaceutical and biopharmaceutical manufacturing. To find out more, please go to mt.com. And now, here's your host for this podcast, the contributing editor for Pharmaceutical Technology, Kim Decker.
Hello, everyone. This is Kim Decker, contributing editor for Pharmaceutical Technology, and I'm here with Dr. Tyler Gable. He's Market Development Manager of Biologics and Biopharmaceuticals at METTLER TOLEDO AutoChem. And I've also got Olivier Berteau. He's Pharmaceutical and Biotech Industry Manager at METTLER TOLEDO Process Analytics, and Dr. Carmen Geisen. She's Pharmaceutical Segment Market Manager at METTLER TOLEDO Industrial Weighing. Thank you all for being here today. Well, let's get started. What are the significant changes in bioprocessing trends, Tyler?
Well, we've seen the pharmaceutical industry as a whole is shifting from mostly small molecule to a greater number of large molecule candidates and emerging technologies such as cell and gene therapies. In this transition, we see many similarities to the experiences of small molecule chemists, which have been learned and refined over about the last 30 years. We all understand that manual process characterization, sampling, and modulation are tedious and laborious, if not impractical without a direct eye into the process. However, bioprocess scientists are really just starting to recognize and embrace many of the same learnings and engineering best practices from small molecule chemistry. This has created quite a significant shift in the technology being applied to bioprocessing. We also see that various interests in continuous processing or other forms of process intensification are mandating heightened levels of understanding and control. But control and the ability to make decisions in real time, when and as needed, is often only made possible from an overlay of multiple orthogonal process parameters and quality attributes. New technologies are constantly being evaluated to provide such insight without requiring such intensive manual process involvement as common with other legacy methods.
What are the typical challenges that labs face when developing and scaling up bioprocesses?
Well, first, this begins with context of historical investments into upstream bioprocesses, which have yielded significant improvements for biomass and tighter outputs over the past decades. But the consequence of this has placed growing burden and now attention to downstream processing operations. This is from both the perspective of the efficiency for the workflow, but also a perspective of the need to retain drug substance yield and quality. There's also a common type of challenge which we see all across the upstream and downstream workflows. That is, when we do try and measure a process parameter or a quality attribute, we may not actually be measuring the direct attribute, rather a surrogate, or might actually be measuring a parameter with an imperfect or inconsistent correlation to an outcome. I frequently do see this problem around processes such as filtration during harvest events. For example, turbidity is a useful parameter in the right situation, but fails to discriminate between particle size and distribution of a population, which is the actual parameter that often correlates to filtration efficiency and product and process quality. A second example that comes to mind is UV-Vis. This is without a doubt a critical and relatively inexpensive technology to provide information on concentration. And UV-Vis will always have a place as a bioprocess measurement, if not from the virtue of the information quality, then at least from the regulatory mandate. However, other methods might be better suited to make a complementary measurement of the process in situ or of multiple components simultaneously, also without requirement for additional analytical methods or sample handling and preparation with greater precision and accuracy than that of a default method such as UV-Vis.
Well, Olivier, we know that inline analysis is always at the junction of process science and scale-up performance. What are your thoughts on this?
This is a true statement technically. A biological model ex vivo is first affected by culture environment. So if only considering external parameters as temperature, mixing, pH, or simply availability of oxygen to feed a cell population, without looking at byproduct accumulation as dissolved CO2 or generation of unwanted oxidative stress, then the scale-up performance will remain performant accordingly to bioreactor ratio and external parameter I've just mentioned.
And in your experience, what are the critical considerations in terms of how inline analysis can impact a fermentation or a cell culture process?
Well, bioprocessing literature is well documented, and all chemical engineering related aspects to optimize mixing, the impact of temperature gradients, the impact of a pH drift, or the aeration strategy are well described and continue to be studied. But beyond those important aspects, accumulation of byproducts, poor dissolution and availability of specific nutrients, and the physiological impact of aeration on cell population and media is becoming, as it is in vivo, a logic path to explore for scientists.
Well, Tyler, how does inline PAT improve bioprocess development and production?
Well, fundamentally, PAT is about delivering higher confidence, constant insight, automation, and time savings where possible. If you can have an automated reactor station make an entire DOE set of samples for you overnight, why would you not? PAT, especially spectroscopy, is commonly talked about relative to controlling a process. But PAT can also just help ensure that you never lose sight of the process to begin with, especially in combination with trends from other biophysical sensors. An in situ spectroscopy method can measure a process continually and identify a root cause deviation through overlay of process parameters where long investigations would otherwise be required, essentially getting to the answers faster.
Why should bioprocess scientists use PAT?
Well, when done well, PAT enables data-driven decisions to be made faster. PAT can often be viewed as complex, but METTLER TOLEDO strives to give any scientist the same power and insight of an expert within minutes. PAT can also be seen, ironically, as more time-intensive, especially when starting out and adopting a new method. But most bioprocess PAT pioneers actually end up realizing significantly improved productivity and effective process development. This is because when applied correctly, PAT acts like a force multiplier. Often incredible amounts of data and higher quality information can be gleaned from fewer experiments and leveraged across an organization. And when you take the time to measure everything, you rarely miss anything.
Olivier, what analytical techniques does the industry need to develop so that we can move from a batch methodology to continuous manufacturing?
Interesting question, but difficult to answer in a couple of minutes. Apart saying growing a cell population in bioreactor or fermenter should better mimic natural environmental conditions. This is specifically valid and important for mammalian, insect, and plant cells. And when thinking of microorganisms, it's almost similar. So it's not about a technique or a specific critical parameter. It's a complex model involving many oxidoreduction chain reaction for cells, influencing metabolic reactions and culture environmental conditions that are playing a major role for cell population and the expression system. So, for example, instead of thinking pH, temperature, or pressure alone, it's a more complex chain monitoring and control model that will certainly help to develop continuous manufacturing models upstream. For downstream, it's a bit more complicated technically to perform continuously a batch and release continuously material for next operations.
Well, now, Carmen, METTLER TOLEDO is known for weighing, and weighing plays an important role in several bioprocessing steps. Can you advise our listeners on how to ensure that their weighing equipment is fit for purpose in bioprocessing?
Weighing equipment can be selected based on a risk assessment and process tolerance through a good weighing practice recommendation performed prior to the equipment acquisition. In addition to scales, a PROEC-specific weigh module accuracy statement is made available from the local sales representative.
Well, how do we ensure that the equipment maintains its accuracy?
This is performed through a series of recommended routine tests and calibration events determined by the scale selection, considering the risk and the overall production process tolerance.
And how do I collect weight data in my control system and ensure that the data is valid and accurate?
High-speed weight data is transmitted over Ethernet or industrial Ethernet to your DCS, PLC, or server. A variety of functions are available, such as alibi log, synchronization, data OK, heartbeat, and alarms aligned to NAMUR NE107.
Okay, well, I'm going to open this up to all of you right now. And if each of you in turn could share one piece of advice with our audience on what's the most important thing they should consider as it relates to your area of expertise and scale up, what would that be? Let's start with you, Tyler.
Well, I would recommend that PAT is not as complicated as you think. Recent advances in technology have made PAT smaller, faster, and significantly easier to use and applicable to a wide range of applications. For example, it's a long persisting myth that infrared does not work in aqueous environments. But especially with proper sampling technology, such as an ATR fiber probe, this has not been true now for many decades. And in fact, has many benefits to offer bioprocessing workflows.
And Olivier?
That's a good question, actually, because when considering improvements of a scale-up or a scale-out model, the most important in order to maintain batch to batch the same quality attributes and productivity expected is the characterization methodology required at scale to better monitor byproduct accumulation and better retro control the impact of byproduct forming and accumulating. So tuning process control in order to accelerate growth rate, eliminate oxidative stress will improve the expression and reduce impact of scale if such methodology to improve performance is applied beyond process understanding and process characterization.
And Carmen, how about you?
Weighing is a critical parameter to ensuring consistent quality. Condition monitoring and real-time diagnostic functions are vital to maintaining the accuracy and continuous functionality of a weighing sensor. Smart load cells embedded in floor scales or in weigh modules signal malfunctions in real time and ensure productivity by avoiding risks and downtime caused by out-of-specification batches.
Wow, everyone, we covered a lot today. I want to thank you all for that informative overview, Tyler, Olivier, and Carmen, and we really appreciate your being here today. This has been Kim Decker, contributing editor for Pharmaceutical Technology, and thanks to all of you for listening.
You've been listening to the Pharmaceutical Technology Podcast, Trends in Bioprocessing Analytics, Weighing and Scale Up. This podcast is brought to you by METTLER TOLEDO, a global leader in industrial analytics, laboratory equipment, and industrial weighing for pharmaceutical and biopharmaceutical manufacturing. To find out more, please go to mt.com.